Hi everyone, welcome to my class. Today we're going to talk a little bit about protein synthesis and specifically the differences in transcription and translation between prokaryotes and eukaryotes. Now these are different cells, don't forget. However, they still contain DNA, genetic material needed for the cell to reproduce. So let's take a look. All right, so today the targets are going to be the student needs to understand the purpose of protein synthesis. So we'll go over that a little bit, but we mostly want to focus on comparing and contrasting transcription and translation in these two organisms. So firstly, let's define what protein synthesis is. All right, and basically speaking, it's the process of making new proteins for the cell. All right, and there are two steps. There's transcription, where the mRNA is copied from the DNA, and then translation, where the actual protein can be produced. And don't forget, DNA is like its own language. All right, it's like translating a word. Maybe you know a word in your language, but you don't know in another language. All right? So the cell needs to take DNA language and transform it into an amino acid language. From amino acids, we can, of course, make the protein molecule. First, it's important that we make sure you know a bit about the differences between a prokaryote and a eukaryotic cell. Eukaryotes can be defined as the type of organism that is either animal, plant, fungi, or protist. All right, single-celled, multi-celled, it still needs to contain these three things. It must have membrane-bound organelles. These are organelles that are bound to the cytoplasm inside the cell. You need to have DNA within the nucleus. All right, DNA is still a constant in any living thing, but we have to have a nucleus for eukaryotes. And there are many other differences, one of which would be that they happen to be larger size than a regular prokaryote. So when we look at the prokaryote side, we see bacteria and archaea, all right, two types of simple single-celled organisms. Well, the differences are pretty much the opposite as a eukaryote. We don't have any membrane-bound organelles. We have DNA, but it's in a nucleoid region. It is not actually contained within a nucleus. And of course, bacteria are much smaller than a regular eukaryotic cell. So remember that. Okay, let's take a look real quick at the transcription and translation in a prokaryote. We'll explain this. We'll also compare it to the eukaryotes on the next slide. Well, first of all, if there are no membrane-bound organelles, this has to all take place within the cytoplasm, all right? The liquidy gel substance that surrounds the entire inside of the cell. And remember that transcription and translation, it doesn't need to go from one place to another. You're already in the cytoplasm, okay? So transcription and translation will actually be coupled together, all right? There will be no breaks, so to speak, as we'll see in the eukaryotes. There's typically no RNA processing. Once the RNA is copied from the DNA, it will have minimal processing, which we'll see in a minute with the eukaryotes, and introns, a non-coding region that is seen in complex eukaryotes DNA, is not present in the bacteria. And we think, well, bacteria is pretty simple, right? Yes, they are very simple. They are not eukaryotes. Okay, now let's compare what we just saw on the last slide to transcription and translation in eukaryotes. So take a look at this diagram for the eukaryote. All right, you can see a lot more happening. Let's go back, compare that to the prokaryotic cell. There's not a lot happening in there, but there is a lot happening in the eukaryotic cell. There's many, many membrane-bound organelles. So transcription will be the first step of protein synthesis, just like in eukaryote. However, in this situation, in a eukaryote, it's going to happen in the nucleus, right? So the DNA cannot be carried out of the nucleus, right? It's going to stay in there, but the cell still makes an mRNA copy. So the transcription copying of the DNA happens in the nucleus, whereas translation would be occurring in the cytoplasm, specifically on a ribosome, the protein synthesis organelle. So what about the time frame between transcription and translation? Well, there's a bit of a lag there. There's a bit of a lag from the mRNA just leaving the nucleus because post-transcriptional modification has to occur. There needs to be further processing to make sure all of the mRNA is ready. There's several things that can be added to the mRNA and removed. All right. So introns, in this case, will be present on the mRNA. There's going to be non-coding regions that are for specific purposes that a eukaryote cell would need and the organism itself would benefit from. Okay, let's summarize this real quick. Bit of a long text here, but I want you to compare an animal, a plant, etc., with a simple bacteria. All right, I'm sure you've seen animals and plants. They are quite complex. How many proteins do you think they have? How many proteins do you think a prokaryote would have? Okay, so in that case, we got to think eukaryotes are very complex. All right, everything from large animals to large trees, small trees, mushrooms, other fungi. They are more complex. They need to be able to produce more proteins, and they must produce them very accurately and completely. So there will be checkpoints along this transcription translation process to ensure that all of the specialized cells, all the proteins that make up these specialized cells, are correct for this complex organism. But take it to another level. Look at the efficiency of the prokaryote. It can move very quickly. We already know bacteria can reproduce very quickly. So what we see is that prokaryotes need to be able to move quickly, responding to change in their environments in a very, very quick manner, whereas an animal may not need to react so quickly in their protein process, but they still need to be more complex. So with a simple structure of no nucleus and no breaks in this process of transcription and translation, it allows for very quick moves, very quick changes in a bacteria. All right. I hope you enjoyed this lesson. Let's take a quick look at this quiz. Take a look at the questions on the screen. Think about the answers. And that's it. Goodbye.